Hello, everyone. Today, we're going to break down the DNA replication process. This presentation was prepared by Dr. C. Helen from the Department of Home Science at Fatima College. Introduction. So, what exactly is DNA replication? Simply put, it's how a cell duplicates its DNA before it divides. This ensures that every new daughter cell gets an exact copy of the genetic blueprint. In complex cells like ours, this happens in the nucleus, while in simpler bacteria, it happens in the cytoplasm. It's a highly accurate process that moves in both directions from a starting point, and it's vital for life, because even a small error here can lead to a mutation. Semi-conservative replication. How does the copying actually happen? We use the semi-conservative model. Back in 1958, scientists Messelson and Stahl proved that when DNA replicates, the original parent ladder splits in half. Each new DNA molecule keeps one original strand and adds one brand new strand. As you can see in the diagram, this hybrid approach is what keeps our genetic information stable generation after generation. The enzymes. Finally, let's meet the molecular machines, or enzymes, that do the heavy lifting. Helicase. Think of this as the zipper. It unwinds the DNA helix. SSB proteins. These act like anchors to keep the strands from snapping back together. Primase. This creates a small starter called a primer so the process can begin. DNA polymerase III. This is the main builder that assembles the new DNA strand. DNA polymerase I. This one is the editor. It removes the starters and fills in the gaps. Ligase. And lastly, ligase acts like glue, joining all the small fragments into one continuous, solid strand. Helicase, the zipper. Think of DNA as a twisted ladder. Helicase is the zipper. It moves down the middle, unzipping the ladder into two separate strands so we can start copying. Primase, the starting line. Next is primase. The builders can't start on their own, they need a starting line. Primase lays down a tiny starter piece so the next enzyme knows exactly where to begin. Polymerase, the builder. Now comes polymerase, the main construction worker. It grabs new building blocks and matches them to the open ladder. It's incredibly fast and even double checks for mistakes as it goes. The fork, the big picture. All of this happens at the replication fork. One side of the ladder is built in one smooth piece, while the other side is built in small chunks. At the very end, a glue enzyme called ligase stitches those chunks together to give us two perfect, identical DNA ladders. The 5 steps of replication. Unzip. Helicase opens up the DNA ladder. Start. Primase marks the starting line with a small primer. Build. DNA pol III adds the new building blocks to the strand. Clean up. DNA pol I removes the starters and fills in the gaps. Glue. DNA ligase stitches everything together into one solid piece. Conclusion. In short, DNA replication is a perfectly timed team effort. By using the semi-conservative model, every new cell gets one old strand and one new strand. This keeps our genetic code stable and accurate. Specialized enzymes like helicase and polymerase work like a high-speed construction crew with almost zero mistakes. Understanding how this works is the key to modern medicine and biology. Thank you for listening.